So here's another concept that we have to understand. It's called alternative splicing. Alternative splicing really just means that there are different patterns, different common patterns that can be formed because of the genes on a hnRNA. And for example, we could have a promoter region at the 5' end of a gene, or of a sequence, and therefore RNA will begin at different. We don't know where this can change where RNA will begin and where it will end. And there's another possibility that we could have exons in the 5' and the 3' regions, and this will also have different impacts. So for example, exons at the very end, and sometimes we have exons being accidentally taken off as well. So this just creates a variation, and there's just this is called the idea behind this is shuffling exons, and it just increases a complexity of how genes are expressed. So for example, we have a hnRNA and it has an intron A, B, C, D, E. There are different possibilities of how splicing is going to cut and modify or fix this hnRNA. It could leave the A, B, C, D gene on it, or it could probably, like in this example, it's cut off the E, or it could even do A, B, C, E, A, E. There are multiple different combinations that are possible. The next question I want to ask is once splicing has occurred, is the RNA or the mRNA, a mature mRNA, ready to be translated? You might be thinking that this is the only one step needed for eukaryotic translation or the eukaryotic mRNA to be ready, but that's not true. There's one step that needs to be done. One more thing. So yes, we have spliceosomes and we have splicing, and the introns are the intervening sequences are deleted. They're gone. So that's good. Whoops, they're gone. So the next thing that happens is we have the exons that are joined together and they're ready to go. But the last thing that happens is at the 5' end of the mRNA we have methylated guanine nucleotides added, and this is called a cap. And you'll understand why this happens in later videos, but at the moment we're just going to memorize this. At the 5' end we have a cap, and the cap is a methylated guanine nucleotides. And then we have the 3' end. We have strings of several hundred never mind. At the end, at the 3' end, we have a string of several hundred adenine nucleotides. This is called a poly-A tail. And this has a different function. We're going to talk about that later on. So here's a question to ask, and this is really going to tell you if you understand how translation and how everything works. If you get this question right, I think you have a good understanding of what's happening. Which is added first, the cap or the poly-A tail? What do you think? And here's a reference to the 5' and the 3' end. And remember, I was screaming in the last couple of videos about something. Well, here's a tip. Let's give you a tip. What direction does transcription proceed in? Well, the answer to that question is the 5' to 3'. So of course, the 5 is added first. The cap is added first, and the poly-A tail is added at the end. So cells often have free-floating exonuclease. Why is that? Well, that's because mRNA has to be degraded, and so here's an example of why we have caps. One good reason is because these viruses have their own kinds of caps and poly-A tails, and the cell has the ability to recognize that this is the mRNA from the cell, or if it's a intruder mRNA from a virus. So this is why we have this is kind of a random question, but free-floating exonucleases that will degrade these mRNAs later on. So now you should have a solid understanding of what transcription is in the first place, and how transcription is different in eukaryotes and prokaryotes. And now you should also understand what hnRNA is, heterogeneous nuclear RNA, and we have overall discussed what transcription is. We have initiation, elongation, and termination, and that really summarizes the whole transcription videos. And remember this thing right over here, and that's when we have transcription on one template of a gene, compared to the other, they're going to make different amino acids at the end.